everyone. This is a continuation of chapter 27, which was all about prokaryotes, bacteria especially. So if you want to go ahead and advance, you should already have notes on these slides. And so this is where I'm actually going to start. These are examples of prokaryotes that were pretty successful at very specialized jobs. So an example of an aerobic prokaryote, meaning when you see aerobic, you're hopefully thinking very good at cell respiration. Notice it has infoldings of its inner plasma membrane where the, little electron transport chains would be built in there. So an example of this would be a proteobacteria, just as the cyanobacterias that we've seen a couple different times this semester have infoldings of their plasma membrane that have electron transport chains built into them that are really good at doing the light reaction. So that's why they're photosynthetic. So the belief is, according to endosymbiosis, that these prokaryotes were free-living, but they would have been engulfed by a larger prokaryote. And when I say engulfed, they're not eaten and destroyed like a foreign object usually is. They were allowed to coexist and live inside, hence the endo. But really, this is an example of really a mutualistic relationship, a plus-plus relationship, where now, for example, if the photosynthetic prokaryote is engulfed inside the larger prokaryote, it can continue to specialize and just do photosynthesis for that host cell. In the meantime, probably gaining some protection and shelter and nutrients. And so really, this is what we know now today as chloroplasts. And if you think about cell respiration, making ATP, these are what we know when these were engulfed by larger prokaryotes. That's what gave rise to our mitochondria. So this slide here should be a review for you. The prokaryotic genome has a lot less DNA, usually just a single circular chromosome, okay, which is actually what most of this is here. Okay, and we know that it's not in a true nucleus, it just has a nucleoid region and where it hangs out. But if you take a look, this particular prokaryote has some very small circular pieces of DNA, and we know those are called plasmids. Keep in mind, these plasmids are not, necessary for basic needs and survival. It's really some of the perk DNA, some of the perks. So, for example, maybe they have genes there for antibiotic resistance. And obviously in there, if that is passed from one bacteria to another, the plasmid has instructions for how to make that sex pilus that it's used during conjugation. So again, not all bacteria have plasmids, just a few. So again, on this page here, remember binary fission is a form of asexual reproduction, and which means that it's going to give rise to clones. Exact identical clones, unless there's some mutation that takes place. And prokaryotes, some prokaryotes have what's called an endospore. And so basically what that means is if there's some kind of harsh environmental conditions, the basic essentials kind of get sucked in to the center of this prokaryote. So for example, just the basic, things like DNA, ribosomes. And so basically it goes dormant. But these endospores can actually regerminate if the environmental conditions improve, and then all of this stuff basically breaks out of this dormancy. It's basically like a hibernation. And these endospores are very difficult to kill. And so we found some, it says here for centuries, but we've actually found some that have been dated to be millions of years old. So this seems like a contradiction compared to the next slide, knowing that prokaryotes do binary fission and their offspring are clones. How would we have genetic diversity? Well, they do reproduce quickly, and remember that there's kind of a trade-off. The faster they reproduce, the more mutation that there's going to be. So mutation could give rise to some new traits, some new novel traits. And if those mutations become more prevalent, we know that it must provide some kind of benefit to the prokaryote genome, and that's why they persist. And then we know that through genetic recombination, prokaryotes are constantly really swapping and exchanging their DNA. They're somewhat promiscuous. And so these are the three ways prokaryotes share. So since they're not doing sexual reproduction where they're actually mating, these are the three ways prokaryotes share or swap DNA. So we know from Griffith's experiment, he did transformation of bacteria and caused pneumonia in those mice. So in that case, it's basically just, you know, a naked piece of DNA that's taken up by bacteria, which we did in our lab. So you came up and I gave you a drop of plasmid, and through the heat shock step, that DNA just kind of snuck in there. So transformation can provide new traits to prokaryotes. Transduction is the image I always have in my mind for transduction is basically where you have a virus. Remember, that's a bacteriophage, or just a phage for short. And here's the bacterial cell. And in that case, remember that viral DNA will enter the cell and incorporate itself into that bacterial DNA, that host DNA. And so now, through transduction, viral transduction, the bacteria has new DNA. Okay, and again, that will be passed down to all of its offspring when it does binary fission. The last one, conjugation, remember, is where we have that male cell that has that plasmid, transfers that over to a female cell, okay, or a negative cell, and now that that female cell has a copy of that plasmid, it incurs a sex change, so it is now a female or a plus cell. Again, notice it's not a mutual swap, so it's a one-way transmission. Again, this slide, remember, you should see prokaryotes in all four of these categories as far as how they get their nutrition. Okay, so the last little bit of this PowerPoint is really classifying bacteria and archaea. So bacteria we divide into 5 clades. Remember, proteobacteria are those aerobic bacteria. They are gram-negative. So remember, what color would those be? Those are going to be pink when stained. And so here are a few of the examples of proteobacteria. Rhizobium is what's pictured here, and these are the soil bacteria that really provide a lot of benefits to soybean farmers. So soil bacteria, these are nitrogen fixers that develop a pretty neat mutualistic relationships with legumes like soybeans. Agrobacterium is another soil bacterium that we use quite a bit in genetic engineering. This is the one, it almost looks like it's forming some kind of a tumor on a plant because they are so good at infecting plant cells. That's why genetic engineers have grabbed onto this technology, because they can use agrobacterium to deliver a gene to the cells that they are infecting. So if you go to Ledges State Park, you will see a lot of plants there, a lot of trees that look like they have tumors on them, and that's because of agrobacterium that has created those. Again, E. coli is one of our gut bacteria, but then also it's a great bacterium that we can use in genetic engineering. And then the last one pictured here is Helicobacter pylori. Again, when you see pylori, pylorus, you should be thinking stomach. And so this is the one that we've known for a while, maybe the cause of some stomach ulcers, and then more recently even some cancers of both the stomach and the early part of the small intestine. So those are all examples of proteobacteria. The chlamydias are, I mean, the name kind of gives it away. Chlamydias are the number one most common sexually transmitted infection in the U.S. They infect animal cells, as we can see here, and the specific chlamydia trachomatis causes blindness. So again, the name is going to give it away. So when you see this, think sexually transmitted. The spirochetes, again, the name is going to give it away. This is, remember, the three most common shapes. One of those is spiral. As you can see here, there's a spiral inside there. And so they are helical. They are heterotrophic, so they need to consume their organic compounds. And there's a couple examples here. One, this is another sexually transmitted. This is the one that causes syphilis. I know that that's spelled wrong. This one here, if you see this characteristic bullseye rash, you definitely want to go and have some testing done for potential Lyme disease. The good news about Lyme disease, because it is bacterial, not viral, it can be treated with antibiotic. And so the most characteristic symptom of that is that bullseye rash. Again, the cyanobacterias, these are all ones that we have looked at or you have Googled over the semester. So these are the photoautotrophic ones that once they were able to generate oxygen, that's going to definitely change the environmental atmosphere. So if you kind of take a look at these and maybe pause your recording, try to name them. So hopefully you recognize, again, they're all going to be green. Don't learn them by color. But Nostoc is the witch's butter. It looks kind of cryptic when you see colonies of Nostoc. Again, the ones that kind of look like filaments of grass are the Oscillatoria. We've also looked at Gleocapsa earlier in the semester, so you'll see them kind of in a colony with this kind of mucilage sheath wrapped around it. You'll see a similar sheath here, but they're really kind of nice and tidy, which is why I like Merismopedia. So it kind of reminds me of an Excel sheet, the way we have, you know, kind of the columns and the rows going across. Okay, so those are cyanobacterias, all photosynthetic. And the last clade of bacteria, we call them gram-positives. So remember, gram-positives, what color would they be? They are purple. And so here are some examples. Some of the names are going to give things away. So Bacillus anthracis is the one that causes anthrax. Clostridium botulinum can cause the food poisoning botulism, which we've definitely diluted that toxin, and so here she's getting a Botox injection. I think your book says something about 1 gram of this toxin could paralyze 1 million people. So obviously they have really super diluted this. But again, it's a neurotoxin, so it's going to paralyze this patient's face, and that's what's going to relax her wrinkles, and that's what she's paying for. Okay. So the actinomycetes is the strain that we get penicillin from. And then probably the most common, that's in the air around you right now, found on shopping carts, found on your skin, we have Staphylococcus, we have Streptococcus. Again, these are the genus names, not the specific species. So, for example, Staphylococcus, we would capitalize. We would capitalize Staphylococcus, and then the species name, epidermis, that's the safe one that's on your skin, usually doesn't cause any problems. But if someone had like Staphylococcus aureus, and it happens to be resistant to methicillin, which is normally a pretty powerful antibiotic, that's what we call MRSA. Okay, so again, these genus names, we'd need to know the species to know more about them. So then the archaea, again, this should be review for you. When you see phyllic in there, you should think that they are a lover. And so halophiles are what are seen in this beautiful salt lake, so they love high concentrations of salt. We also have lovers of temperature here. So for example, the thermophiles living in the hot springs that helped us simplify PCR. And then here, gens means you're creating something. And so these are methane creators. Okay, so, living in the guts of some animals. They live in swamps. Okay, so again, those archaea are pretty cool prokaryotes because they really live in some pretty unique environments. So this chart here, it can be kind of overwhelming, but if you look at it in comparison, so for example, if you're looking at the archaea, if you count up how many similarities there are with bacteria, okay, here's 1, 2, really 3 similarities between the bacteria and the archaea, and then you compare that with archaea and eukaryotes. Again, looking down these 2 columns, I'm going to put a check mark anytime I see some similarities. Okay, so if you take a look here, we've got 1, 2, 3, 4, 5, 6 similarities between archaea and eukaryote. So the conclusion should be that archaea are more similar to eukaryotes than they are to their prokaryote bacteria. And that's really why archaea got its own domain, because we used to actually classify bacteria and archaea together. Okay, so more similar to archaea than to the bacteria. Okay, and that's kind of surprising. We wouldn't expect that. So this, just to wrap up, these last few slides deal with kind of the big picture about prokaryotes. Even though that we know they can be pesky and we have to run to the doctor to get an antibiotic for our strep throat, or maybe we have a stomach ulcer caused by Helicobacter pylori. But they really do play crucial roles in the earth. And so some of them are decomposers. They might recycle nitrogen and make it available in a form that plants can't do on their own. They could be, cleaning up the oil spills, making our environment a little cleaner from all the things that humans do to impact it. In a smaller situation, we know, even within our own body, we have normal, residents, we call it resident flora, resident life of bacteria in our intestine that's basically munching on our leftovers, the things we eat that we can't digest, and really making vitamins available to us. So remember, mutualism is a plus plus where we both benefit. So those intestinal flora are getting a nice, warm, dark place to live, and a few of our leftovers in exchange for those vitamins. Commensalism is a plus zero relationship, and that would really probably be our staph epidermis that, is munching on those dead skin cells, which isn't harming us. It's not really helping us, unless, maybe that's your source of hygiene for bathing if you're dependent on it. But for the most part, they're not harming us. So it's a plus zero. We're not getting really anything out of that deal. Parasitism, of course, we know is plus negative. So I've got a few examples of some disease-causing prokaryotes on the next slide. But again, this is showing the rhizobium, which is a mutualistic, a plus relationship with soybeans. Here, of course, this fish has a bioluminescent bacteria that's living on its side here, and that's probably helping that fish attract its food, its prey, and then while it's eating and munching away, there's probably a few leftovers left behind for those bacteria. So plus. Okay, so here's some of those disease-causing or pathogenic prokaryotes. They usually, one of the things that disease-causing prokaryotes are capable of is by either releasing an exotoxin from their surface, or they contain a toxin inside their cell wall. Okay, so an example of this, again, remember the Botox, the botulism toxin is an exotoxin, whereas, if you eat some raw eggs or some raw chicken, and you actually have to ingest the actual salmonella bacteria in order to get the endotoxin. It's still a part of its body. And so one of the issues, of course, since 9/11 is the risk of bioterrorism. There's been several anthrax scares where individuals in government have been mailed envelopes full of spores, and people, if they inhale that, they get a respiratory form of anthrax that is very highly fatal. It can't be contracted from person to person unless they're opening that envelope together and both breathing in the spores. But if you see like hazmat teams arriving in their suits, they're protecting themselves while they investigate to see if they truly are anthrax spores. And then here are some of the positives that we've talked about during the semester. There's a few new ones here. So bacteria can be used to make plastic. We know that we use a lot of bacteria when we're making, for example, antibodies for infections, hormones, synthetic vitamins. Bacteria are small, they're quick, they're easy to grow, and so they're wonderful for DNA technology applications.